Welcome to QuantaLife. I'm Bill Colston and I'm the CEO. I'm here to talk to you about the team and the technology that's developing one of the first DNA diagnostics for viruses such as the H1N1 flu. We started developing this technology 5 years ago at one of the nation's premier research laboratories. At the time, we were trying to rapidly detect bacteria and viruses. It took hours to days to make this measurement. Here at QuantaLife, we're reducing that down to minutes. For the last 20 years, Lawrence Livermore National Laboratory has built the country's largest biological defense program. We established the science and reduced this technology down to practice. Now the team and the technology have come together to form QuantaLife to commercialize this product and bring it to market. We are QuantaLife, and here's how we do it.
My name's Isaac Bright, and I'm Vice President of Molecular Diagnostics here at QuantaLife. My goal is to align our strategy around developing molecular diagnostic kits that provide very high clinical utility. We have proprietary oil chemistries, proprietary thermocycling systems, and detection systems that, when working in concert, allow us to provide nucleic acid testing on a scale that's never been done in a time frame that's never been seen before. Currently, when most systems are operating with a central lab surveillance system, it takes at a minimum 6 to 8 hours if they're providing polymerase chain reaction testing, oftentimes 2 days if they're working with a culture system. With our system, you're talking about having definitive results on the order of 10 to 15 minutes. So there are many tests on the market now for agents such as methicillin-resistant Staph aureus or vancomycin-resistant Enterococcus. And our main goal in competing in those particular spaces would be to bring those tests to the point of care so that you had meaningful results from the test from that particular patient in the time that it took to triage the patient. This system will be a very easy system to use. Your typical nurse or technician should be able to operate it at or near the point of care. When you think about the need to respond to infectious outbreaks, you need to be able to have definitive data about a patient's status so that you can put them in the right place and start immediate therapy appropriately. The course of their infection will then be much less. You've decreased exposure to other patients that are adjacent. You've decreased exposure to healthcare workers because in total, the course of their infection is much shorter.
My name is Heredia, and I'm in charge of assay development. My role here is to streamline the process of developing new test kits. A new test kit can easily be integrated into our platform by simply changing out the cartridge, and this is very analogous to the way an inkjet printer cartridge fits into a printer. The facility we're in right now here at QuantaLife is a biocontainment facility, so we're capable of developing new assays for a wide variety of viral or bacterial pathogens. We can develop a new test kit in about 2 months, and if there's already an assay out there, we can rapidly adapt it to our system.
So my name is Amy Hiddessen, and my role is to ensure that the internal chemistry is reliable and robust. The idea of the system is to make faster, smaller reactions using microdroplet technology. A microdroplet is a mini reactor that contains a single copy of DNA, which can be from a virus or a bacterium. We know how much material we're starting with in the reaction, therefore the analysis is quantitative. Droplets are made in an inexpensive disposable cartridge. Inside this cartridge, we're able to make droplets that all have the same or uniform size. We have an internal quality control process whereby we watch the stability of the droplets through the device. We're looking for any changes or instabilities in these droplet reactors as they go through each step in the reaction. Each droplet can be thought of as an indestructible test tube. And the advantage of making individual droplet reactors is so that we eliminate the possibility of cross-contamination.
My name is Masquelier, and I'm a senior design engineer here at QuantaLife. My role is to take basic concepts that people have come up with and to gear towards the final product. Our concept of a thermocycler is much different than conventional PCR. In conventional PCR, you have to take a fairly large volume which adds up to high thermal mass, and you need to raise and lower the temperature of that volume, which requires a fair amount of time. In our system, because we have microdroplets, we can change the temperatures very rapidly. So what we do is we take small curved pieces of metal, and on the backside we actually put a heater. And we make up a segmented device, and then the capillary tube is simply wound around the outside of the metal. As the droplet is transitioning from one region to the next, probably within a second it's to the proper temperature. So there's very little time wasted in transitioning from one temperature to the next. So the technique of doing multiple tests in a quanticore is quite simple. And once you've moved the first batch of sample through, at that point you can add a slug of oil that becomes the separator from the next sample. There's very few parts and very few moving parts.
My name is Kevin Ness, and I am initial founder. I'm currently the Director of Research and Development. What I do here is basically make sure that every droplet counts. The droplets, after being thermally cycled for a few minutes, are put back in single file and pass through the detection site. The droplets move past a laser beam, and as you can see now, each flash of light is analyzing a single droplet. On the scope, each one of these spikes is information of what's occurring within the droplet. The large spikes correspond to positive droplets that initially started with DNA. The smaller spikes are droplets that did not start with DNA. So once they're monitored, I take the ratio of positives and negatives, and that gives us a concrete answer of exactly the starting sample concentration. My job is to make sure that the results are accurate and reported in simple, actionable terms for the clinician.
My name's Ben Hindson. I'm the Chief Science Officer here at QuantiLife. My role is to oversee the scientific development of our diagnostic pipeline. So develop our kits and our instrument and get it in the hands of our customer as quickly as possible. The instrument is designed to be a point-of-care type instrument, so it's the size of an inkjet printer, runs off standard power, and can generate lab quality results in a matter of 15 minutes. So tests that typically take days to run, you can get those results almost instantaneously. The device is very simple to use. All you have to do is take a swab from the patient, break that swab off into our disposable cartridge, and then essentially downstream from that, the instrument does the rest. So it takes the bacteria or virus, breaks it open, releases nucleic acid, washes it, and then concentrates it into a very small volume. That volume then is converted into droplets within the cartridge. Those droplets then flow very quickly through a capillary tube. In that capillary, they're heated and cooled very rapidly, which enables billions of copies of DNA to be made. After amplification, the droplets flow single file through a fluorescence detector. That's exciting it with a laser beam, and it's measuring how bright each droplet is. And then that gives us the amount of DNA that was present in the sample and tells us whether or not, in fact, that harmful bacteria or harmful virus was present in that patient's specimen.
From a simple swab test, we can get laboratory quality results in less than 15 minutes. That's what this technology is all about. We are going to transform the way doctors and clinicians treat patients.